first step is glycolysis. In this step, a glucose is broken down into two pyruvic acids or pyruvates. So, begin with a glucose, which is a 6-carbon compound. It uses 2 ATPs to break up glucose into two 3-carbon compounds called glyceraldehyde triphosphate. And before we get to pyruvic acids, 4 ATPs are produced. As you can see, it has to invest some energy in the beginning in order to activate the process, even though the purpose of this process is to generate ATPs. Back to before we get to pyruvic acids, 2 NADH are also produced in this step. Therefore, the total gains from this step are 2 ATPs and 2 NADH. Moving on to the next step, which occurs in the matrix, the inner compartment of mitochondria will focus on only one pyruvic acid. But before a pyruvic acid is ready to be used in the next step of cellular respiration, it needs to be oxidized, meaning that one of the carbon is cleaved off of the pyruvic acid. Also, NAD+ is reduced to NADH and carbon dioxide is produced. So, you end up with 2 carbon compounds called acetyl-CoA, one NADH, and a carbon dioxide. Now, the acetyl-CoA merged to a 4-carbon molecule called oxaloacetic acid, which I have to apologize that I forgot to write it down in the video. However, they form a citrate, which is a 6-carbon molecule. Citrate becomes another 6-carbon molecule, cis-acetate. Then, cis-acetate becomes isocitrate, another 6-carbon molecule. In the next step, we lose one of the carbon in a form of carbon dioxide, and we also produce one NADH. So, we left with a 5-carbon molecule called alpha-ketocoutarate. The same thing happens again. We lose another carbon as carbon dioxide and produce one NADH, but this time we left with a 4-carbon molecule known as succinyl-CoA. Next, it produces GTP, which is equivalent to ATP. Then, the succinyl-CoA is converted to succinate. After that, succinate converts into fumarate. Fumarate becomes malate. And FADH2 is produced. In fact, FADH2 is only produced in this process of cellular respiration. Moving on, malate becomes oxaloacetate, completing the cycle. It also produces another NADH. The cycle is called Krebs cycle, which is the second step of cellular respiration. This is the last step of cellular respiration, which is electron transport chain and chemiosmosis. In eukaryotes, the electron transport chain contains series of electron carriers that are located in the inner membrane of the mitochondria. And in prokaryotes, it is in the membrane. Both of them have the same goal, that is to generate ATP. High energy electrons, which are NAD+, forming NADH, and then enter the mitochondria. Then they join with NADH and FADH2 by Krebs cycle. High energy electrons are passed from one carrier to the next. During two high energy electrons move down the electron transport chain, they also transport hydrogen ions across the layer's membrane. So, how hydrogen ions come from? In this case, electron transport chain changes FADH2 to FAD and NADH to NAD+, where hydrogen ions are removed. In the end of electron transport chain, oxygen is an important factor that helps combination low energy electrons and hydrogen ions, which are waste, to produce water or H2O. High energy electrons move back to the chemiosmosis or ATP synthesis to produce ATP. By movement of hydrogen ion generates ADP to ATP.